Hi, uh, we continue with lipid metabolism, talking about fatty acid synthesis and the formation of tri. I will go through very quickly just a reminder of the different classes of fatty acids, and then we will talk about in details about fatty acid synthesis, substrate energy, cofactors, mitochondria and cytosolic involvement, enzymes and regulation, modification of the fatty acids, and finally the formation of the storage form of the fatty acids, which is triacylglycerol or triglycerides. Again, if you wish to read the words for this particular lecture, you can find the word file in the Basic Metabolic Pathways folder on Entrada. And I found you a very short video to look through fatty acid synthesis if you wish to see someone else's lecture for more understanding. So here we are in fatty acid metabolism. And you may recall that we went through the triglycerides broken down to fatty acids and glycerol. Glycerol is going to the glycolysis or gluconeogenesis process, and fatty acids are going to the mitochondria in the form of acetyl coenzyme A after beta oxidation to feed into energy generation. Now we will look into the opposite direction, that when we synthesize fatty acids and then we store away in the form of triglycerides. So fatty acids are long chain, short chain, or medium chain apolar species, which is a carboxyl group to give a little polarity to the fatty acids. We have saturated and unsaturated fatty acids, which means that this carbohydrate chain may or may not contain double bonds. We also have branched and unbranched fatty acids, and we also have essential and non-essential fatty acids. Just a reminder that linoleic and linolenic acid are very important for our well-being. These are essential fatty acids we need to take them up from food, and they are the precursors of arachidonic acid, this one, arachidonic acid, which is the starting material for icosanoid prostaglandin leukotriene thromboxane synthesis, which you already heard a lot in this curriculum about in this curriculum. So this arachidonic acid is actually coming from linolenic acid. So you see here the double bonds. These are the unsaturated fatty acids. We cannot position the double bonds into the essential fatty acids. We just cannot synthesize that. But we can synthesize other unsaturated fatty acids, and we label them with fatty acids with the number of the carbons. So for example, this is palmitic acid, which has 16 carbons and no double bond. So after the colon there is a 0, it means that it is a saturated fatty acid. If after the colon you have a number, for example in palmitoleic acid, you know that you have one double bond, and the 9 in parentheses tells you what position it is, actually starting from the carboxyl group back. So it is in the number 9 position. Now, this omega-3 and omega-6, which are linoleic and linolenic acid. So let's say just omega-3, it is 18 carbon, 3 double bonds in the position of 9, 12, and 15. So you see that we can put a double bond into number 9 position, but we cannot put double bonds into 12 or 15. This is why we call them essential fatty acids. Now, this 12 is actually, and 15, the 12 is actually 6 carbons back from the other terminal of the fatty acid, and the omega-3 means that this 15 is 3 carbons back from the other term. That is just for fun. You don't need to remember that, but if you will ever work with fatty acids and you question whether it is a C12 position or it is an omega-6 position, then you know that recently we count the carbons from the carboxyl group. In more ancient times, the carbons were counted from backwards. All right, let's go further. This is to show you that we have short chain, medium chain, long chain, and very long chain fatty acids. These are the carbon numbers going with that. Why do I emphasize this? Do you remember that when we talked about beta oxidation of the fatty acids, a lecture before or a case before, we emphasized that these medium and short chain fatty acids do not need carnitine to go to the mitochondria for beta oxidation. So it's important to know that although we do synthesize long chain fatty acids, and this is what we do usually for storage purposes, when it is necessary, we can also synthesize medium chain fatty acids. Actually, the site of this is the mammary gland. And the very long chain fatty acids, which already, again, you heard about, very important for the central nervous system, needs a different beta oxidation system starting in the peroxisomes and continues in the mitochondria. So these are the short chain, these are the medium chain, and these are the long chain fatty acids, and from 18 and up is actually very long chain fatty acids. And you see that the essential fatty acids actually are long chain fatty acids. This is just a note for your future. If any of your patients who is becoming a mother and kind of thinks about not to breastfeed the kids, you can talk about the nutritional value of the milk for newborns and infants. So related to this lecture and the previous one, short and medium chain fatty acids are rich in milk. Actually, there is no long chain fatty acid there. And these are the ones that can directly absorb from the enterocytes and go to the circulation right away, rather than packaged in chylomicrons and going, in a longer way to feed us. They go directly to the portal bud and they go directly to the mitochondria. They don't need carnitine to take them up and go for energy generation in mitochondria. Milk also contains essential fatty acids that are transferred from the mother's diet, mother blood, to the milk. There are short peptides. There are no long proteins that need to go through a long digestive process in the small intestine of the baby, but these are short peptides that can be easily digested by the surface peptidases of the mucosal cells. They also contain essential amino acids from the mother's diet and the mother's blood, so the baby will be well fed and can synthesize enormous amount of proteins that is needed for the baby to live and to grow. Now, it contains glucose and not sucrose. Again, there is no need for digestion. It can go right away for the portal blood and can be taken up by any cell for energy. And the milk also contains essential vitamins, minerals. It also contains antibodies. If I am not mistaken, these are IgAs. So everything is there in the mother's milk, in the mammals, not just in human, to feed the newborn and feed the youngs. So just if someone is interested more in it, it's interesting to understand why breastfeeding is so important for babies. Let us go back to the fatty acids and triglycerides. So after fatty acids are synthesized, which we can do a lot, Then they are esterified, glycerol esterified with 3 fatty acids, and this is the storage form of the fatty acids, as you already heard many times. So let's look into how we synthesize fatty acids. Generally, we synthesize palmitic acid, which is C16, which can be then elongated for longer carbon, more carbons. It can be modified. It can be desaturated for unsaturated fatty acids. It can be further modified with branches, with hydroxylation, etc. So I will focus on the synthesis of palmitic acid, C16. So what are the main steps, which I will talk about later? First of all, acetyl coenzyme A is the building block of the fatty acid. So the fatty acid grows with 2 carbons. Acetyl coenzyme A is produced in the mitochondria, correct? You remember that. So it needs to be transported to the cytosol because fatty acid synthesis is a cytosolic process. So for acetyl coenzyme A synthesis, we use glucose, and then we transport out this acetyl coenzyme A to the cytosol to produce fatty acids over there. Now, you may recall what the transport form of acetyl coenzyme A is. Do you? If not, I can tell you it is citrate, and I will show you in the next figure. The acetyl coenzyme A needs to be activated to be able to enter the fatty acid biosynthetic process. And the activated form of acetyl coenzyme A is actually malonyl coenzyme A. And the enzyme which is doing that, increasing the carbon number from 2 to 3, is the so-called acetyl coenzyme A carboxylase. Fatty acid then is synthesized from malonyl coenzyme A and one single acetyl coenzyme A to start with, and this system is a wonderful system which I will talk about. You don't need to remember everything. Don't worry. This is a multifunctional protein which is called fatty acid synthase. And then the palmitate can be modified further. So the 2 enzymes that I will focus on are acetyl coenzyme A carboxylase and fatty acid synthase. Let's start at the beginning. Fatty acids are synthesized from glucose. So when we eat, glucose comes into the cells, and liver and adipose can use the glucose, the extra glucose, which is in the liver, remember, it's not stored away in glycogen or not used for cholesterol synthesis or not used for other purposes, then it can be used extra glucose to feed to glycolysis, pyruvate going to the mitochondria. Mitochondria is producing acetyl coenzyme A, transported out in the form of citrate. You remember this one? You need pyruvate carboxylase to produce oxaloacetate. You need pyruvate dehydrogenase to produce acetyl coenzyme A, and the 2 needs to form citrate as a very first step of the Krebs cycle, and that this citrate will go out to the cytosol. So when I ask you what is the transport form from the mitochondria to cytosol of acetyl coenzyme A, you will remember this beautiful system which we went through minimum 3 times by now. Okay, so citrate. Citrate goes out. We don't need citrate, we need acetyl coenzyme A. Thus, the citrate need to be lysed to produce acetyl coenzyme A and back oxaloacetate. Oxaloacetate doesn't have the transporter to go back to the mitochondria, but it needs to be reduced to malate, and we use NADH for that, which comes from glycolysis. Malate then can go back to pyruvate, and the enzyme is a decarboxylase, and we need NADPH for that. What is producing NADPH? Nothing else but the hexose monophosphate shunt, which is also feeding off of glycolysis and glucose. Now, so we have the pyruvate, we have now acetyl coenzyme A here, and then we can feed this into the fatty acid synthetic system. I would like you to more or less remember this figure because it contains a lot of important things. One, it's glucose, glucose, and glucose that supplies a lot of things for starting fatty acid synthesis. Also, NADPH, not just for the malic enzyme, but also for the fatty acid synthesis process, which needs a lot of redox steps, which uses NADPH. Now, what we talked about it until now, that for the palmitate, which is carbon 16, we need 8 acetyl coenzyme A's coming out from the mitochondria. We need NADPH, 8 of them, to actually feed into this system. And then this NADPH is produced partially, and this is the 8 produced by the malic enzyme, but the rest should be produced by the hexose monophosphate shunt. Now, so this is what we will do, and I just wanted to show you that here is glucose. Glucose is going to pyruvate, goes to the mitochondria, produces acetyl coenzyme A, and this acetyl coenzyme A needs to go out to the cytosol for malonyl coenzyme A, produce the fatty acids, and then it can be used to further modify to triglycerides. All right, so activation of acetyl coenzyme A to malonyl coenzyme A is a committed step of fatty acid synthesis, which means that when the acetyl coenzyme A is turned to malonyl coenzyme A, it will go nowhere else but being used for fatty acid synthesis. This is the enzyme called acetyl coenzyme A carboxylase, which means that the product will be 1 carbon longer, and the carbon for every reaction when we need to build in carbon, when we want to expand the carbon number with 1, will be a carbon dioxide, which is coming from bicarbonate. This is an energy requiring reaction. We need ATP, also supplied by glycolysis. Now, malonyl coenzyme A is produced, and I have a question here. Which pathway, which other pathway that you learned about involved another carboxylase? So this was pyruvate carboxylase, you remember? Even the previous figure contained that to produce oxaloacetate, and oxaloacetate is important for transferring the acetyl coenzyme A out of the cytosol, but it is also the starting material of gluconeogenesis, and oxaloacetate is also, and pyruvate carboxylase is the anaplerotic reaction for Krebs cycle. So this is the committed step. Committed steps are usually regulated. So how do we regulate this enzyme? Actually, it's very funny and it's very interesting for a biochemist to think about it. Not necessarily you, but I love it. So acetyl coenzyme A carboxylase is a cytosolic enzyme which is a dimer, but it is inactive in a dimer form. It actually needs to polymerize. So this polymerization and thus activation of acetyl coenzyme A carboxylase is regulated by 2 molecules. One is citrate, which is, you remember, coming freshly out of the mitochondria to produce fatty acids. So that's the positive allosteric regulator. And the long-chain fatty acid coenzyme A, which is the end product of fatty acid synthesis, is the negative regulator. Very logical. However, because this is such an important enzyme to start the fatty acid synthetic process, it's also regulated by hormones. Again, no surprise here. When glycolysis, which supplies so much for fatty acid synthesis, is running, it's insulin. So insulin will regulate this enzyme to prepare it for fatty acid synthesis. How is insulin regulating enzymes? Usually through a signaling pathway which produces a phosphatase. It takes off the phosphate from the target enzymes, so the dephosphorylated form of acetylcoenzymic carboxylase is active. When do we want to inactivate it? We phosphorylate it. You remember what the counteracting hormones of insulin are? Glucagon and epinephrine in the liver. It will upregulate a kinase which will phosphorylate the enzyme. Sorry. There is, however, another signal, and this is the so-called AMP kinase. AMP is produced from ATP when there is lack of energy in the liver, and that AMP is activating AMP kinase. AMP kinase is inactivating the enzyme. So what did you learn? This enzyme, which is the committed step for fatty acid, is regulated by allosteric regulators and regulated by phosphorylation, dephosphorylation through different hormones and lack of energy, AMP kinase. This table is actually interesting to read because it brings in other point of view than just the strictly biochemical point of insulin up, glucagon down, allosteric regulators work. It brings in actually an important measure that we need to always think about. What if we are on a high carbohydrate diet? What if we are on a fat-free diet? What if we are in a high-fat diet or fasting or well-fed? So these actually are nothing else but dysregulation, but from a larger point of view. So if you are on a high carbohydrate diet, obviously in insulin will regulate. However, if you stimulate the liver cells with insulin through high carbohydrate diet, the enzyme synthesis, so the acetylcoenzyme A carboxylase gene will be upregulated and more enzyme will be in the cytosol. So what is the consequence? You are getting fat. Sorry to say that. We are producing more fatty acids, we are producing more triglycerides, we are sending it out to the circulation, and we are depositing in the adipose tissue and happily gaining weight. Now, what if we on a fat-free diet? Well, we need fatty acids. So we will synthesize fatty acids through, again, upregulating the enzyme synthesis, more fatty acid synthesis, more fatty acid production. What if we are on a high-fat diet? Then we will decrease the availability of the enzyme because enough fat is coming in, so why on earth should we synthesize it? Fasting also decreasing enzyme synthesis because if we fast, we should not synthesize fatty acids, we actually use fatty acids for energy. And in the liver, we use fatty acids for energy to supply energy for gluconeogenesis. Right? So think about it a little bit. It again is important to understand how the metabolic processes are integrated in one cell, but also how it is integrated in the whole body. Right, so we will have the activated acetyl coenzyme A, and now we feed this into synthesizing fatty acids, growing by 2 carbons. So this absolutely wonderful, beautiful, great system putting together the C16 from 2 carbons is called fatty acid synthase, which is a multifunctional enzyme, which means that all of the activities that are needed to produce fatty acids are actually within one polypeptide chain. What do we have there? 6 enzymes and one special unit which we call acyl carrier protein, which will actually let the growing fatty acid chain go from one enzyme to another to elongate and elongate and elongate the fatty acid chain. And it is not enough that we have one big protein which has 6 enzymatic catalytic centers and one carrier protein center, but we have 2 of them working together and feeding into each other. So it is a very, very efficient system to produce fatty acids. And this enzyme, this multifunctional enzyme, is not letting go the fatty acid until it reaches the 16 carbons, when actually the last activity of this enzyme out of the 6 will hydrolyze off the ready fatty acid from this multifunctional protein. Absolutely beautiful. So you don't need to memorize these steps, but let me guide you through this process to understand how the fatty acid grows and to understand where and why do we need so many NADPH molecules. So the very first step, as I said, for the growing of the fatty acid, we need malonyl coenzyme A, but actually the very first step is an acetyl coenzyme A binding to the enzyme. Then the malonyl coenzyme A is added. Then we lose one carbon dioxide. That carbon dioxide that was put on the acetyl coenzyme A to activate it to be able for these 2 molecules to fuse into now a 4-carbon residue. This already looks like a modified fatty acid, but it is not yet a fatty acid, correct? We have an extra carbonyl group here, which we need to reduce. So NADPH is used. We have hydroxyl here. Now we take out this water. This is a so-called dehydratase. And then we have a double bond. We don't need this unsaturated fatty acids to start with, so we need to reduce it again. And look at that. We have a very, very similar 4-carbon short-chain fatty acid here. But you remember, this multifunctional protein enzyme is not letting go the fatty acids until they are actually 16 carbon long. So this will be repeated, and other malonyl coenzyme A comes in. We lose the carbon dioxide. We have now 6 carbons. Reduction, dehydration, reduction. Now we have a 6 carbon fatty acid still bound to the enzyme, the multifunctional one. And then we repeat until we have 16. And then the last activity of the enzyme is actually taking this fatty acid off from the fatty acid synthase. And this is this beauty here. This is just amazing. So this is the ACP, the carrier protein, and we have 1, 2, 3, 4, 5, 6 enzyme activities, and the growing chain is going through the different enzymes and elongated and elongated and elongated, and the final step is actually taking off the long chain fatty acids from this. Beautiful, isn't it? We also regulate fatty acid synthase. The allosteric activator is nothing else but glucose 6-phosphate. Again, you remember, glucose comes to glycolysis, very first product is glucose 6-phosphate, and it already sends the message to this enzyme down the road that be active because I am putting acetyl coenzyme A out and malonyl coenzyme A out for you to synthesize fatty acids. Again, insulin upregulates the activity, the transcription, and glucagon downregulates the transcription. So there is no phosphorylation, dephosphorylation of the enzyme. And the same thing that I showed you before, if you want to think about diet and what the diet, of course, through hormones, will do to you, whether you will synthesize more or you synthesize less fatty acids, it is here again. Now, let us calculate how much does it take to synthesize a palmitic acid. I'm telling you, it's very expensive. So if we add everything together, we need 7 ATPs and 14 NADPHs, a lot. So let's count. Again, you don't need to remember every step, but here is the 1N single acetyl coenzyme A to start the synthetic process. Then we have 7 malonyl coenzyme A's. And when we push through this particular molecule, through the synthetic process, we need 14 NADPH. However, the malonyl coenzyme A came from acetyl coenzyme A, and we utilize 7 ATPs here. So if we add everything together, we have the 7 ATPs, the 14 NADPH, and it is a very expensive synthetic process. However, it worth the energy to be put into this molecule, because you remember, when we need this energy and we go through the beta oxidation of this one single palmitic acid, we have 131 ATPs gained from that molecule. Now, very little that I want you to remember. I really want you to remember that we can elongate these fatty acids. This is especially important in the central nervous system because we need those very long chain fatty acids there. It is an enzyme, multifunctional enzyme, which is similar to fatty acid synthase, and it can be located either in the ER or in the mitochondria, depending on which system we are talking about, which cells. We can desaturate and we can hydroxylate these fatty acids, and we use mixed function oxidases which work with NADPH and oxygen. So what is this enzyme? Nothing else but CYP, cytochrome P450. It's an ER function. We can produce shorter fatty acids only in the mammary gland. We can take them up from milk products, and we can produce it in the mammary gland during the lactation period. And I just wanted to remember that how important short and medium chain fatty acids are for infants and children. We can produce branched chain fatty acids. Methylmalonyl coenzyme A will be used in that step instead of malonyl coenzyme A. Now, let me finish very quickly that we produce fatty acids. Again, remember the two cells that can do that are liver and adipose, especially liver. But we don't release fatty acids from the liver. We actually produce triacylglycerols, and we package them in VRDL, which goes out to the circulation and releases to the adipose tissue for storage or for other tissues for energy generation. Again, adipose tissue is the storage facility. It can store a lot of fatty acids, up to a month or even 3 months worth of energy, but there is a short half-life for these adipocytes. They are actually turning over fast, synthesizing, taking when it is insulin signal and releasing when it is glucagon or epinephrine signal. And this is just a picture of the fat cell, how much triacylglycerols can be stored in the cell by everything is pushed. All the nucleus, the mitochondria, the ER, everything is pushed to the side of the cells. And you can see it also here. Fat cells very full with triglycerides, and this is the pure little nucleus pushed to the side of the cell. So how do we synthesize it? We need to activate the fatty acids to fatty acid coenzyme A's, and then we react them with glycerol. Glycerol is activated to glycerol 3-phosphate. So let's see how it goes. So in the adipose and in the liver cells, it's the same. Glucose goes through glycolysis and through the glycolytic process produces glycerol phosphate. The difference between the adipose tissue and the liver cell is that glycerol can be taken up by the liver and it can directly be phosphorylated to glycerol phosphate. So we have the active glycerol in the form of glycerol phosphate, and we have the activated fatty acid in the form of fatty acid coenzyme A, and they will react with each other. One fatty acid is bound to the glycerol phosphate. We call it lysophosphatidic acid. Then another fatty acid will be esterifying glycerol, and we call it phosphatidic acid. And I would like to stop here for a second. This phosphatidic acid can be used to produce triacylglycerol, with actually removing the phosphate and esterifying the glycerol with a third fatty acid. But depending on what the liver cell wants to do, this phosphatidic acid also is the starting material to produce all kinds of phosphoglycerides, to build the membrane, to react with, to have the bioactive function of the phospholipids, and so on. Now, this is something important to remember. This is a direct interplay between two different pathways. One is glycolysis or glucose catabolism, and one is fatty acid synthesis. One is glucose catabolism from glycolysis and also feeding to the hexose 1-phosphate shunt, and glucose supplies everything for fatty acid and triglyceride synthesis. I hinted it in the previous couple of minutes, but let's summarize it. Glucose supplies acetyl coenzyme A, which is the starting material for fatty acid synthesis. Glucose supplies NADH, which is used for and necessary to the acetyl coenzyme A successful transport and utilization in the cytosol. Glucose supplies NADPH through the hexose 1-phosphate shunt. Glucose supplies the energy what we need for fatty acid synthesis in the form of ATP. And finally, glucose supplies glycerol phosphate. Is it not amazing? So, what did we go through today? Fatty acids come from different form. Please remember that we have saturated, unsaturated, short chain, medium chain, long chain, and very long chain, branched and unbranched. Did I say saturated and unsaturated fatty acids? C16, what we synthesize in the liver, and then we modify it further. Please remember, we need to eat essential fatty acids from food. We cannot do that. You cannot synthesize that. So starting material, acetyl coenzyme A comes from carbs. Glycolysis in the cytosol. Pyruvate goes in. Acetyl coenzyme A is produced in the mitochondria and also carboxylated and comes out from to the cytosol, where it is carboxylated and activated to malonyl coenzyme A. Please remember the enzyme and its regulation. Then the multifunctional enzyme of fatty acid synthase takes over and it produces the palmitic acid. So acetyl coenzyme A, malonyl coenzyme A, NADPH, and for this step, ATP is needed to synthesize fatty acids. Malonyl coenzyme A and fatty acid synthase, both of them are regulated. Please remember how. Palmitate then can be further modified, and we can produce longer or hydroxylated or unsaturated fatty acids from them. Shorter fatty acids are produced in the mammary gland during lactation period. And we can package this newly synthesized fatty acids in the form of triacylglycerol in the liver, adipose. From the liver, it goes out to the general circulation in the form of VLDL. Thank you for your attention.